Our next speaker is Dr. Terry Hyman-Peterson. Dr. Hyman-Peterson is Professor of Neurology at the Louise Katz School of Medicine at Temple University, where she is Director of the Center for Neurodegenerative Diseases and for the MDA ALS Center of Hope. She's board certified in neurology with subspecialty certification in neuromuscular diseases. Her research has focused on ALS and includes both clinical and laboratory interests. She's president and co-founder of the ALS Hope Foundation, which is a nonprofit committed to making a difference to people living with ALS. She's committed to patient care and education, actively participating in the Clinical Research Learning Institute to train PALs to be research advocates. So Dr. Hyman-Peterson is going to be talking to us about genetics, providing insights into ALS disease mechanisms and targeted treatments.
Hi, my name is Terry Hyman-Peterson, and this is a real privilege to be here to talk to you about the genetic landscape of ALS and how it has helped to inform us on disease mechanism and inform us on developing treatment strategies. Today, we're going to talk about all of this, as well as cell-based and animal models of ALS. So 10% of ALS is familial. What do we mean by that? It means that there's a family history of someone else in the family having had the disease. The first gene that we identified was the SOD1 gene in 1993 that, when mutated, caused disease. Approximately 20% of familial ALS can be explained by mutations in the SOD1 gene, and there have been more than 187 different mutations found. Of import is that even 1 to 2% of sporadic ALS folks can harbor a mutation in the SOD1 gene. This gene also shows complexity and variability even within families, so that within a family where the SOD1 gene tracks with disease, one can see two family members with the same mutation, but yet having very different clinical disease, different onset, and different survival. This suggests that there are other factors that modify disease, including genetic factors and perhaps environmental factors, as well as aging. Certainly, this has provided the nidus from my research, which is in the genetic modifiers of ALS in animal models and helping to translate that to people. And I hope to have some time to talk about that at the end of this talk. Well, we got SOD1 in 1993, but the discovery of genes has really accelerated over the last 10 to 15 years. And you can see that in this graph where just from 2008 on, it's been an explosion. And now more than 42 different genes have been identified that are associated with ALS. And this accounts for more than 70% of the genetic component of familial ALS. And why have we had such an acceleration? It's really been due to technological advancements. The first thing was the sequencing of the human genome in around 2000 led to the ability to really database and data query on changes that were in the genome that could be tracking with ALS and where they were located and in what genes. The sequencing speed, how fast we could sequence somebody's genome and how cheaply was one of the next advancements. The use of different techniques like genome-wide association, whole exome, and whole genome sequencing, and comparing affected and control individuals, and the speed that we could do that, and the improvement in statistical techniques applied to those methodologies, all have helped to really accelerate. And just in the last 5 years, there have been 11 genes identified. If one looks at the distribution of the genetic changes in familial ALS, you can see that we can identify a genetic underpinning and what particular gene is affected in almost 70% of cases. I also want to point out that in 10 to 15%, this says 11%, we identify genetic changes in the sporadic population where there's no family history. The most common of all of the genetic defects has been the C9 open reading frame 72 gene. In this gene, it isn't a change in the sequence of the coding region, but rather an extra piece of DNA inserted in uncoded region. That is, regions of the DNA that don't necessarily translate to the working protein C9orf72. But as a result of this increased piece of DNA, there ends up being translated proteins called dipeptide repeats that end up glomming up the cell. They affect nuclear transport, nucleocytoplasmic transport, and they also cause aggregation within the cytoplasm. And there's a possible toxic gain of function. And it does make up 40% of familial ALS, and importantly, 4 to 7% of sporadic ALS. And if we can find a treatment for C9, we will be able to help virtually 15 to 20% of all ALS. Think about that. And so understanding these genetic changes and getting therapeutic approaches is going to be extremely important, not only for familial ALS, but for sporadic ALS. Similarly, you can see the distribution of some of the other genes that have been identified. And I should comment that different populations have different distributions of genetic abnormalities. To add to the complexity, so we have 42 genes. We have families where even within the same family, the same mutation can look very different. And here we see that there are families that actually harbor two different affected genes, two different mutated genes, so that clearly there's even influence between those genes. We've also identified a gene called ataxin-2, which can modify the presentation of ALS. Ataxin is also a repeat disease where there's an increased piece of DNA. If that piece of DNA is big enough, you get an ataxia. But if it's only increased moderately, it can alter the way ALS looks. And in fact, there are some treatment strategies for people who carry that extra piece of DNA in their ataxin gene. There's treatment strategies that are directed at that as well. So it's a very complex interaction, and the more we understand, the more we'll be able to direct treatment both for sporadic and for familial ALS. To add to all of this, ALS is on a continuum with other neurodegenerative diseases, one of which is frontotemporal dementia. And there's a relationship here. 40 to 50% of people with ALS have cognitive involvement. It can be very mild. 5 to 10% have frank frontotemporal dementia. And it turns out that ALS and frontotemporal dementia not only share pathologic changes, specifically protein aggregates with mislocalized TDP43. This is a very important protein. In a small number of familial ALS cases, there's actually a mutation, but in virtually all ALS folks, be sporadic or familial, there is clumping or aggregation of this protein, and the protein is mislocalized in the cell. It's supposed to be found in the nucleus, but it ends up in the cytoplasm and clumped. That same pathologic finding is seen in FTD, again, a shared pathologic change. Furthermore, there are genes that are shared and can result in either FTD or ALS. And probably the most common one that we pay attention to is that C9 gene, but certainly FUS, TDP43, And you can see a whole list of them that we don't have to list or mention. I just want to impress that there is a huge overlap, even genetically, between these two diseases. And we come up with this whole picture. It's very complex. I think Dr. Hardiman alluded to that ALS is an interplay between your genetic endowment, aging, and your exposures over time, probably some chance. So very complex, and the more we understand, the more we'll be able to design appropriate treatments and even prevention. But I always like to say that the genetic changes in ALS have led to an understanding of motor neuron biology gene by gene. And there's also been targeted several important pathways that these mutated genes play a role in, implying that these pathways are important for the pathophysiology of ALS and as targets for treatment. So, we have the nucleocytoplasmic transport, where things go in and out of the nucleus, and C9 is probably the most important gene to remember there. We have the way RNA and DNA is really metabolized and handled. And again, TDP43, FUS, and C9 are important there. We have protein aggregation, or the way proteins are handled and discarded and metabolized. That there's a huge number of genes which when mutated cause ALS and also play a role in protein homeostasis or protein aggregation. Neuroinflammation is yet another area of import, and certainly there are certain genes which play a role here. TBK1 is probably one of the most important ones, but also MCT and TREM2 play a role in pathways of oligos and microglial function, which bears on support cells and neuroinflammation. And of course, we have mitochondrial function and oxidative stress. SOD1, our first gene ever identified, has a toxic gain of function which increases oxidative stress, but also it turns out C9 and TDP43 play a role here. And we all know that we have some newer drugs that target oxidative stress. We also have several drugs in trial that target neuroinflammation. Another area of import is the vesicle transport and axonal transport, as well as the cytoskeleton of the nerve cell. And so here you can see several genes that disrupt cytoskeleton and axonal transport, and several that are target vesicle transport defects. All of this important for neuronal function. And finally, excitotoxicity. So these give us several different areas that we can target. And if you think of them and condense them, it's neuroinflammation, it's RNA, DNA metabolism, protein homeostasis and aggregation, and really oxidative stress and mitochondrial function. And finally, vesicle transport, axonal transport, and cytoskeleton, that grouping of pathophysiology. So let's talk a little bit about treatment in familial ALS and how it informs treatment. I've already alluded to the fact that these mechanisms can be targeted. We can study the mechanisms in animal models and iPS cells and mimic the disease and try to modulate the change. That doesn't necessarily mean it's going to do the same thing in people. We typically think of two major mechanisms: gain of function, which generally tends to be a gene that has a mutation that's dominantly inherited, meaning that you only need one damaged gene. Remember that we inherit pairs of chromosomes and genes. One set of our chromosomes comes from mom and one from dad. And so that in we have 2 copies of every chromosome and 2 copies of every gene. A chromosome has all the genes, like a necklace of pearls, each pearl being a gene. In autosomal dominant disorders, you only need 1 bad gene or 1 damaged gene. And these tend to be a toxic gain of function pathology. Loss of function is typically recessive. That's where you need 2 damaged genes. And octoneuron and TBK1 are examples of recessively inherited genetic abnormalities. We have identified toxic gain of function and a way to treat toxic gain of function by using strategies that will decrease the amount of protein made, so that you decrease the amount of toxic protein. However, there's a caveat here, because by decreasing the protein, you may also decrease some of the function of that protein. And there's some indication that while, for instance, SOD1 and C9 are predominantly toxic gain of function mutations, there's also some loss of function. And if you further decrease function, there may be actually ramifications that you don't want when you treat someone. So we need to be cautious as we develop these treatments. And of course, in toxic gain of function, just to review something that I think you've already heard about, we use an antisense strategy where we use typically DNA is made into RNA, which then is translated into protein. By providing an antisense oligonucleotide, it will bind the RNA and prevent it from becoming protein and rather cause it to be degraded, thereby reducing the amount of the theoretically toxic protein. And this strategy is being used with SOD1 and also with C9, where the antisense is actually directed at that increased area of DNA that doesn't even code for protein, but codes for these dinucleotide repeat proteins that glom up the cells, theoretically. And in FUS. In addition, I mentioned that ataxin-2, which modifies disease if there's a little bit too much DNA in that repeat region, ataxin has repeats, there's been an antisense strategy directed at that to see if it can modify disease in folks who carry that increased expansion. So moving on to ALS models. These models can be cell-based. These are simple models. They can look at the function associated with mutated genes or the effect those genes have on proteins, and also be a way to perturb cells and test drugs in a rapid throughput manner. And these include cell lines, budding yeast, and importantly, human-derived iPS cells, which gives us a human-based system. And in fact, there's been some evidence of reproduction of phenotype and pathologic change in iPS cells, so that they do show shortened survival, for instance, and they do show aggregation of proteins. However, it's not yet been shown how well they translate to human disease and inform us on treatment strategies, but that work is ongoing. In addition, we can have small animals like C. elegans, the worm, zebrafish, flies. These are really nice because they're easily genetically manipulable. So you can put in a genetic mutation, take it out, put in 2 mutations. They're relatively inexpensive compared to mouse and larger models, and rapidly can be used in genetic screens to identify mutations, and also to look at the consequences of those mutations and how to manipulate them. And larger animals more closely mimic human disease, more complex, and we can study pathogenic mechanisms and proof of concept. The promise of translating clinical effect of a drug in an animal model to people has not yet been proven, but we've been asking a model for SOD1 disease to do that, and I think that's almost unfair to the model. That model, however, was used in early antisense development and showed an effect, and we're hoping that that does translate, because that's a SOD1 disease to a SOD1 disease. And this just is to impress upon you all the different mutations that have been placed into these model systems and can be used for these types of studies. And as you can see, everything from cell culture, iPS cells, to mice and rats. And so I'm going to talk briefly about rodent models as we finish up. There have been rodent models made for many of the genetic mutations, especially the more common ones, SOD1 being the first and probably the most commonly used. And they really are a gold standard for preclinical ALS modeling of disease, not necessarily for translation of clinical effect. And we use them to study disease mechanisms, and they've certainly taught us a little bit about motor neuron degeneration. And in the SOD1 mouse, this is an overexpression model where we put mutated human SOD1 genetic material into the animal model, and we do multiple copies, 23 copies. The more we put in, the worse those animals are. And, you know, as I mentioned, this animal model led to understanding gene silencing strategies using ASOs. For me, I have exploited this model to look for genetic modifiers. And it turns out that if I put that mutated human gene in different strains of mice, there's a different phenotype. The mouse has either a milder disease or a more severe disease. So on the background of SJL, there's a milder disease, more severe disease rather, with earlier onset and shorter survival. Whereas in a black 6, there is a later onset and longer survival. We used this or leveraged this information to actually link the disease to a mouse chromosome in chromosome 17 in collaboration with Jack's laboratories. Both of us independently identified this chromosomal location and worked to narrow the region. And once the region was narrowed, we then were able to identify what genes were in the region and could possibly play a role. And the way we do that is to identify the genes in the region, to look at the sequence in the mild versus the severe phenotype and see where there are differences and where those differences would result in abnormal protein, an abnormal protein, and also which ones were previously reported to be important to motor neuron biology or ALS. We have shared that list, and I'll talk about that in a moment when I talk about translating this information to people. But in addition, we wanted to make sure that this was not only important for SOD1 ALS, but more universally applicable. And so we looked at another animal model of motor neuron disease, the dynactin model, and in point of fact found out that yes, background did matter. And again, that SJL white mouse had a shorter survival, more severe disease, while the black 6 background had a milder disease. And we again went on and we performed linkage studies and recently have linked this also to chromosome 17 and a similar region, suggesting that the modifiers will modify motor neuron degeneration in general and not be specific for a particular mutation. We've shared the genes within these regions with our collaborators. We have shared it with Brian Trainer at the NIH so he can look at his GWAS studies, his genome-wide association studies for signals from our implicated genes in the ALS population versus controls. And we've also shared it with Molly Fetnani and her collaborators at the New York Genome Center, where they can examine DNA from ALS folks correlated with phenotype, the severe versus a milder phenotype for our implicated genes to see if they also could be modifiers in humans and in people, as well as looking at CNS tissue from ALS folks that's segregated by phenotype, more severe, earlier onset, shorter duration versus later onset, longer duration, to see if there's any difference in the expression of our candidate genes. And we're hoping that we will translate what we've learned from the animal model to people. In conclusion, ALS is genetically complex. There are genetic influences in familial and sporadic ALS both, and up to 11-15% of sporadic ALS carry mutated genes. And there are other influences besides just having that gene that influence your clinical picture. And these genes will continue to inform us about the pathophysiology of ALS, provide models for studying diseases, and direct treatment opportunities both for genetic variants and sporadic. And we have to really understand the mutations and consider both gain and loss of functions mechanisms when we look at the therapy, so we don't harm anyone and so that we produce an efficacious effect and don't knock a gene down so much that we lose more function and make people worse. So it does take caution, care, but the promise is there. And I thank you for your attention.
Okay, great. Thank you so much, Dr. Hyman-Petersen, for that very comprehensive genetic overview.